Thank you for the invitation. First of all, dear chair, dear colleagues, I would like to present our work. It's not my work, just only my work, but it's really a teamwork and cooperation of Hyde Company and the University of Page, where very enthusiastic people work on these topics. So what is the topic? Basically, yes, I can jump. Yeah. So basically, we focused on the clinics and we combined the clinical cohorts data with the animal studies. So we wanted to understand if the deuterium depleted water has a role in the prevention of cancer. Our model is not really preventing the cancer, but studying the effect of carcinogenic molecules on molecular level on the activity of the genes, so that we measure the gene expression. So there are dedicated genes which involved in the carcinogenesis, but they also activated after a carcinogenic exposure. So we use them as a biomarkers of carcinogenic exposure. So in this way, when we studied the deuterium depleted water, it's mainly blocking somehow the effect of carcinogenic chemicals. So this is just a summary of what we've done. So we involved patients, voluntary patients who consumed the deuterium depleted water under the protocol of Hyde Company. And we designed, an animal in vitro model and study. But first of all, I would like to talk about the lung cancer. And you know very well the importance of lung cancer. It's a leading cause of death among cancers. And unfortunately, Hungary is in a very bad situation regarding the lung cancer. We are world leaders. Maybe you did not know this. And I wondered why, because we do not have the highest rate of smokers in the population, but mainly there is a correlation with the diet, consuming smoked food or something like that. So, by the way, we are leaders. So it's a very important issue. And the problem is that people who would reach the retirement age, they develop lung cancer. So the lung cancer appears around 65 years. So exactly when people reach this age. And unfortunately, the survival of lung cancer patients is still very poor. Okay. And the other question is, so most of the people do not live longer than 1 year. And why the deuterium depleted water? Because there was an experience with deuterium depleted water in cancer treatment in animal models, and there were some data, clinical data, which have proven that deuterium depleted water may provide longer survival of people. So on prostate cancer, breast cancer, we had data. So that's why we focused on this additional or supplementary treatments besides the conventional therapy of the cancer. Here is the design of our studies. There was the cohort study with patients. 129 patients were followed, and the majority of the people were men, but or the higher rate of involved patients were men. But you know this very well that mainly men will suffer in lung cancer because of the higher rate of smoking, basically. And we distinguished also non-small cell lung cancer and small cell lung cancer, brain metastatic patients, and we compared, of course, the male and the female survival after this additional treatment. In the animal study, we used the carcinogenic treatment. We used the DMBA. It's a known carcinogen. It causes mutations, and it increases the activity of the genes. The expression of the genes are elevated, and these genes or these expressions can be used as a biomarkers of carcinogenic exposure. So we use this model, and a mouse model. They were young mice, and we treated them. And not just treating with DMBA, but we combined the carcinogenic molecule with the deuterium depleted water. So these animals consumed the deuterium depleted water, and the controls consumed the normal or conventional level of deuterium, what you can find in the tap water. Of course, there was a statistical analysis. Okay, so here is the distribution of patients by tumor type and sex. The first, the males, and then the females. Non-small cell lung cancer, non-small cell adenocarcinoma, and squamous cell carcinoma, small cell lung cancer, and there are the rest of the patients where there was no clear diagnosis. So what we can read here, that the dominant type was the non-small cell lung cancer, and there was some difference between males and females. For example, the small cell lung cancer rate was higher in male population. Okay, yeah. Okay, so when we analyzed the survival rate, we could find that the treatment was beneficial for the patients, and there was difference between males and females. Also, there were age groups, and we could find that by sex, females survived longer, and there was also a correlation with the stadium of the cancer. Of course, with lower grade, people survive longer, and with advanced cancer, people do not have as long survival as with the less advanced stage. So what we could read here is the median survival was 25, close to 26 months in male population, but in females, where the patients drank deuterium depleted water, it could raise to 74 months. And here is the value in months, the 27 months for brain metastatic patients. The longest survival was 87.5 months. So here we can read the different values of the survival, and below you can see the survival, the 5-year survival rates. So the median survival times, what you can read above, in males and females. Surprisingly, in females, it's a huge difference compared to the males and compared to the conventional therapy. It's extremely high here. So you can see the 74 and 87, for example, in non-small cell lung cancer. Also, the brain metastasis, we could reach nice results. You can see here the not calculable in males because we could not measure the survival because males survive longer. So we could not calculate. So this is not a bad result. This is even a good result. So when we analyze the 5-year survival rate, it is much longer than with the conventional therapy. So 19% in males and 33% with living brain metastasis, and living with small cell lung cancer, it is 25%. In females overall, it is 42%. So if we compare the 42% to 19%, so the females and males, so there is a huge difference here also. So if we jump to the animal experiments, sorry for the crowded slides. Maybe you cannot see well. I wanted to overview all the data, but so if you just follow the bars, it will help. The first two. Okay, the first one is just the water, the conventional water with the regular deuterium concentration. The second one with the same water, but with the carcinogenic molecule, the DMBA. So what you can see here is this is the first gene which was studied, the KRAS, and you can see how the DMBA could increase the activity, the expression of the KRAS. So okay, so the water plus carcinogen. Here the BCL2, the next gene, the water plus here the carcinogen, you can see very well the increase. So these were the controls, and the last gene was the MYC gene. So with water and with carcinogen. So it's very clear what happened. And the next two columns with the 25 ppm deuterium depleted water alone, and then with the carcinogen. So just the low deuterium water and then with the carcinogen. What we can see here is that there is no dramatic increase in the expression of the KRAS. So if we compare to the conventional water plus the carcinogen, so if we compare these two, this is the case in KRAS, and this is the same situation with BCL2. And with the CMYC, it is not the same if we treat it with DMBA or not. With low deuterium water, but the bar is far is not high than here when we use the conventional water and the carcinogen. So this was true in female mice. So these are the females, the first. So this is very nice, we could see an effect. But in case of the males, we could not see this effect. Even these mice did not respond too much to the carcinogenic treatment in case of KIRAS, but there was some increase in BCL2 and c-MYC. And we can see the same pattern, more or less, if the mice consume the deuterium depleted water. So these genes, KIRAS, maybe you know the background information. The KIRAS is quite frequently mutated in lung cancer. But here we did not focus on the mutation. We did not focus on the overexpression based on the mutation or overactivity because of the mutation, but this was just a biomarker of the response to the carcinogenic exposure. But KIRAS and BCL2 are involved in the survival and apoptotic functions of the cell. Also, c-MYC, which drives the cell cycle, working as a transcription factor and modifying the activity of other genes. So deuterium depleted water could nicely silence these genes. So this can be the answer. I mean, their activity can be the answer of the carcinogenic exposure, but maybe there is a correlation with the survival of people who consume the deuterium depleted water. It's very crowded. I'm sorry. I am going to summarize our findings. First of all, the survival of people. So we had historical controls. We did not have a control group, but we had statistical data from the Hungarian population. So these people were Hungarian patients, and we compared our results to the general lung cancer patients. So the survival is calculated or compared to the general statistics. So what we could find is that instead of living 1 year, these people lived even much longer. It depends on the subtype of the lung cancer, but in female category, they survived more. The median survival time was more than 6 years. So instead of 1 year. So it's a huge difference. If we focus on the small cell lung cancer, there was also a nice improvement in the survival. So it was clear and statistically significant increase in the survival. But this is true for female population. This is true for women. In the men, it was not significant. So this was the same situation in brain metastasis. People lived much longer after consuming the deuterium depleted water. Then to study a little bit the cancer patients, maybe there was a bias choosing these people who voluntarily consumed the deuterium depleted water. I wondered, maybe this is a special group who already lives longer. And I studied a little bit the supplements, what people take, And 80% of the cancer patients take supplements. And the advantage of taking supplements is it is very little in the survival. So what we could find is just 11%. It's this advantage in the survival. So what we could find with deuterium depleted water, it is much, much harder effect, much, much stronger effect. And if we focus on the mechanisms of action, what I told you that it can decrease the effect of carcinogenic molecules or carcinogenic effects. So we focused on chemicals. So I cannot tell the physical, the results of physical carcinogens, but we focused on a group of chemical carcinogens. So the deuterium depleted water could diminish the effect of carcinogens in our experiment. And we focused on genes which are involved also in generally the carcinogenesis and the cancer formation. So if we silence them in the stage of early carcinogenesis, it is not excluded that there will be a silencing later. We don't know, just I cannot exclude. So finally, the conclusion, what we could find. Just a summary. Deuterium depleted water is non-toxic. It's a natural product. And survival was dramatic in lung cancer patients, especially in females, especially in women population. In mice, we could see that there was the diminishing of the effect of carcinogenic molecule. And additionally, the brain metastasis patients live longer, not just people living with the primary cancer. So there was a clear difference between women and females and males. This is my last slide. I could find some interesting correlation with lung cancer incidence, lung cancer death rate, and the deuterium concentration in the tap water. So where you can see the higher incidence, the darker colors, and the higher death rate, darker colors, and the deuterium concentration in the tap water, the red one is the more concentrated deuterium, and the blue one is the depleted deuterium in the tap water. So if you compare the maps, so this is not a deep study, just an interesting thing that, okay, so it seems that there is a correlation. If there is a deuterium depleted tap water, there are less number of cases and less number of deaths. Okay. Thank you for your attention.
Okay.